Hello, today we are going to learn what about pyruvate oxidation. So what is this process? This is a process which links glycolysis and citric TCA cycle or citric acid cycle. Okay. So in this process, in glycolysis, the final product that we are getting from glycolysis is final product pyruvate. And the initial, like the substrate in TCA cycle is acetyl coenzyme. So we need to convert this pyruvate into acetyl coenzyme. This is what exactly this process. Yeah, so what we were telling that this process, it is simply the conversion of pyruvate to acetyl coenzyme. So it did do contain three enzymes: E1, E2, E3. So the first enzyme is called pyruvate dehydrogenase. Second enzyme is called dihydrolypoyl transacetylase. Third enzyme is dihydrolypoyl dehydro. So this is basically what this is an enzyme complex containing the subunits E1, E2, E3. And this enzyme complex is called what? Pyruvate dehydrogenase complex. This is a complex known as pyruvate dehydrogenase complex. You do have three subunits: E1, E2, E3. E1 is pyruvate dehydrogenase, E2 is dihydrolypoyl transacetylase, and E3 is dihydrolypoyl dehydrogenase. Okay, and what are these functions? They are functions. E1's function is decarboxylation. Decarboxylation. E2's function, from the name, suggests that transacetylase, that means acetyl transfer. Acetyl transfer. And the third step is E3, dihydrolypoyl dehydrogenase, which means regeneration of what? Lipoic acid. Okay, so these are the functions of the subunits of pyruvate dehydrogenase complex. And what are okay, one more thing is that there are some cofactors uh, in these steps. These also you need to study. Okay, cofactors in all the three steps are cofactors in the first step is TPP. Second step is coenzyme A and lipoH. Third step is NAD plus and FAD. So this is a complex which contain five what five cofactors and three enzymes. Okay, so these are the process which contain five cofactors: 1, 2, 3, 4, 5 cofactors and three enzymes. What exactly that happens in this process is, in each step, like in each enzyme, there is two steps happening that you should remember. Let's dive into the steps. So yeah, so the first step is by E1 pyruvate dehydrogenase, which converts what pyruvate to the presence of E1 plus TPP, and in this process carbon dioxide is also released. Okay, in this process it release hydroxyethyl plus hydroxyethyl TPP. This complex is formed. Okay, and this hydroxyethyl TPP in the it will get oxidized. And what will happen? It will convert into acetyl and TPP. So this is the first two steps in E1. You can say that this is the first step and this is the second step. Okay, you can remember it like that. For every enzyme there will be two steps. So the final product in from the enzyme one is acetyl. Okay, and from acetyl, this acetyl will bind to what E2 in the presence of lipoate. What it will form is acetyl coenzyme A. In this step, in the presence of lipoate, a complex will be formed by acetyl and E2 and lipoate, and this in the presence of coenzyme A will form acetyl coenzyme A. Okay, this acetyl coenzyme A. So what are the two steps here? This is one process. Okay, this is one step, and from here this is another step, acetyl coenzyme. So acetyl coenzyme is formed. This is the first product. This is the product which we want to add in TCA cycle. Okay, so the third step is the regeneration. In a regeneration step, this lipoate which will get reduced in this process. The process of converting this into this, it will get reduced lipoate. This reduced lipoate in the presence of FAD to FADH2, there will be 2 electrons, right? And in FADH2, these 2 electrons will convert into NAD to NADH. So in this process, this reduced lipoid will convert to what? It will get oxidized in this process. So here also what you have to remember is that from FAD to FADH2 there is one step, and from FADH2 to there is 2 electrons present, and this electron will add to NAD plus to form NADH. So this is the 2 steps, okay, and it will form acetyl coenzyme A and a oxidized what? Lipoid. So these are the process in pyruvate dehydrogenase complex, and we have seen the cofactors. If you want, I can mention the cofactors once again. In first step, TPP. The second step, coenzyme and lipoid. In the third step, it is FAD and NAD plus. Sorry. NAD plus and FAD. They are the cofactors. Okay. And in this process, there are inhibitors and activators of this process. So inhibitors of this process, first we can write activators. Activators means which activates this process. When is this process needed? When we need energy, when we need ATP. Okay, so if there are a high amount of ATP present, will this process happen? No, this is a negative modulator. So if there is more ADP present, will this process happen? Yes, because ATP is not present. The raw form ADP is present. It is not phosphorylated. So activators, one of the activators is ADP. And in this process, there is the cofactor of coenzyme A, which having it converted to acetyl coenzyme A. So these are activators. If there is a large amount of coenzyme A, the process will get activated. If there is a large amount of ADP, the process will get activated. And if there is a large amount of NAD plus, the process will get activated. Similarly, we can write its products like ADP. When this process happens is ATP is formed. So if there is enough ATP present in the cell itself, we don't need to have this process. Okay, so this is a negative modulator, and coenzyme is acetyl coenzyme A. So the product is itself formed, so we don't need this process. And NAD, the final product is NADH. So we have to study this 3. You have to just remember this 3: activator and 3 inhibitors, and excuse me. Okay, so you have to learn that the complex, this pyruvate dehydrogenase complex, about its cofactors, its function, its activators and inhibitors, and you just need to read and understand what is the process taking place in these steps. So these are all in this topic, so you have to study all of these things. Okay.